So good morning. This is a second session in the biological and pathology overall session. And my name is Hermann-Georg Holzhutter from the Charité, the medical school in Berlin. And I will be chairman and speaker in one person. So I was invited to give an introduction into a new field that emerged about 10, 15 years ago, systems biology. And when I was invited, I was not clear what level, scientific level, will be appropriate. So I finally decided to make it very simple. So in the way I give lectures to my medical students, if you find it too simple, then I have to apologize for it. So let's start. The whole field of life sciences is subdivided into well-defined sub-branches, as for example molecular biology, neurobiology, entomology, and other fields which have all their own subject, their specific methods, their own journals, their own chairs at the university. So this field is well covered, seemingly. So the question is why there is a new field called systems biology. Is it really required? Is it something new? Is it only a new name for already existing fields? So to understand how this field came in, it's maybe useful to have a very brief look back into the history of biology. And the starting point of modern biology was the important observation made in the mid of the 19th century and phrased by two famous German scientists, Rudolf Virchow and Theodor Schwann, who stated that the cell is the basic functional unit of all living systems. So all organisms, whether they are unicellular or multicellular, made out of at least one cell. So the number of cells in the organism can be quite different. So can we somehow remove this bottom line? No. Because it's really disturbing.
Yeah, I'm gonna try. What's the... I really don't understand what's this.
Sorry for this. 1 minute, please. No, it's better, thank you. Let me go back. So whether it is a very simple organism like this worm, this Sinorhabditis elegans, which is a favored object in biological research, has less than 1,000 cells, or it is a human having 10 to the 13 somatic cells, depending on the size of the human, of course. Well, all they are composed of cells. And the cells can only originate from cells. So this was a very important statement. So this is a famous sentence, omne cellulae cellular, that every cell originates from another existing cell. And this is true up to now. There is no way to compose a living cell from organic molecules up to now. Although there are some efforts to do that, but they are not successful. So the cell as such is a very complex system. It is composed of many cellular organelles, like, for example, the nucleus, the mitochondria, the so-called power stations, producing the energy, most of the energy required, the endoplasmic reticulum, and many other organelles, depending on the specific cell type. So it is good at this point to remember that microscopy has enabled the establishment of cell biology as a scientific field, because it gave the opportunity to magnify these very small, these tiny biological objects. And then the Leeuwenhoek microscope, which had only one lens, had a magnification of about a factor of 250. Modern light microscopes have higher magnifications, up to 2,000, 3,000. This is a limitation. And it gave an insight into the overall composition of the cell, but most importantly, it enabled to monitor cell division as a process where two cells originate from one mother cell, so, which was the foundation of this so-called cell theory in biology. And later on, owing to this invention of the electron microscope, it was then possible to look further into the cell, to see the organelles in more detail. And altogether, this gives a very comprehensive view, but it's a static one. You can see only frozen, immobile structures, so it doesn't give any impression what is the dynamics inside the cell. So the question arose, what is the information carrier inside a cell that allows the cell to transmit all of its components into the daughter cell? So what is a building plan, where it is resident in the cell? And for a long time it was believed that these are the proteins, because there is a multiplicity of proteins inside the cell. They form very complex structures. And nobody up to, let's say, beginning of the 20th century thought that is a DNA. The DNA had been the first time isolated and described by Friedrich Miescher in 1866. And it took almost 100 years then to arrive at a reliable model of this molecule by this famous work of Watson and Crick. One should mention that that was a final step, and before many other famous biochemists had already identified the molecules that compose DNA, the base pairing principle had been established by Shagraf. So it was really the final step to present the full double helix model of the DNA by Watson and Crick. And then it became clear that all the information required to build the cell structures, is encoded in the DNA, which can transcribe, not the DNA as such, but enzymes can describe this information into messenger RNA, and this messenger RNA then is bound to the ribosome, and the ribosome is a place where this information, sequence information contained in the mRNA is translated into a protein sequence, which then forms more or less autonomously to a functional protein. The mRNA in a cell has a relatively short lifetime, normally only a few hours. Some mRNAs have a longer lifespan. The proteins may have a half-life of 1 week or even longer, but there are also very short-lived proteins having a half-life of only 4 or 5 minutes, so quite different lifetimes. The proteins can be degraded in a process that we call proteolysis, and this liberates the amino acids as a building blocks, which then can be reused for the de novo synthesis of a protein. So this is, in essence, what we call the central dogma of molecular biology, which is a flow of information from DNA versus mRNA to the protein, and the proteins are the most important functional molecules of the cell, because they are the only molecules that are really encoded, so this information can be inherited. All other molecules of the cell are made up by the action of proteins, and proteins amount only to about 17% of the total cell mass. So that means the majority of molecules composing the cells are secondary products produced by proteins. So it became clear that DNA is the central information carrier encoding for the proteins of the cell. Proteins then make all these organic molecules that are required for the functioning of the cell as lipids, triglycerides, membrane lipids, carbohydrates, which can be monomers or polysaccharides, nucleotides, even amino acids. At least a limited set of amino acids can be produced, and other organic molecules. And they together form the organelles, which then compose a cell, and cells and their interaction form the tissues, organs, and finally the organism. So it was really tempting to understand how this information that is present in the DNA is translated into the phenotype, the organism and its functions. So, and the idea was to start this enterprise by sequencing the DNA and then to identify all genes in a human cell, and then to go on along this line of information flow to translate the sequence information of DNA into what we call the phenotype, with all its properties like aging, susceptibility to certain diseases, and other phenotypic characteristics. So this was the point where it was decided to start a worldwide research project aiming at the encoding of DNA, the full sequencing of human DNA. There was international alliance led by scientists of the United States. Francis Collins, who is the second person on this cover picture, was the leader at this time at the Bethesda, and other scientists and groups in the United Kingdom, France, Australia, Japan, I should also mention Germany, and many other groups took part in this project. And the reason why it was successful is that the techniques available to sequence nucleic acid had advanced, so that they could be automated. There were automated that were able to sequence a large portion of nucleic acids in a very short time. There was another private initiative which was headed by Craig Venter and its company Cellular Genomics. There was some race, some competition between these two consortia, and they published almost at the same time a draft sequence of the whole human genome. The full genome was then published 3 years later in April 2003. So the key and I would say disappointing finding was that we have about 23,000 genes in our genome, which is approximately the same range as in mice and in roundworms. That Our genome contains a significantly higher portion of repeated sections of DNA, so some redundancy in the genome, and that it contains less than 10% of protein families that appear to be vertebrate specific. So the surprisingly low number of genes has to be considered with some caution, because one gene that is defined as that a stretch of DNA that encodes one polypeptide chain, one gene can encode more than one protein. This is due to procedures that appear during transcription, so the so-called splicing, and allows to combine the exons in different ways, and there are many post-translational chemical modifications possible. So in principle, one gene can produce more than 1,000 different proteins, so that is the reason why the number of active proteins in the cell is certainly much higher. So having completed this project, the question was, what can we do with this information? And the work was mostly done based on full genome sequences was to identify genetic markers. So the idea was to look at variations in the sequence composition of DNA from different individuals, and to find clues that could give a hint why this person has a higher risk to suffer from a certain type of disease. For example, having a family where all family members suffer from a heart attack before having, before having reached the normal age, which is typical for this type of disease, then there is a suspicion they must have a certain genetic disposition for this type of disease. And thousands of papers had meanwhile been published reporting on so-called genetic markers, which are variations in the DNA of groups of people knowing to be extremely susceptible to a certain type of disease. And the so-called single nucleotide polymorphism is the main working tool here, because it is known that at specific sequence positions of DNA we observe very frequently a change of one nucleotide, of one base. And this gives a typical pattern. And each individual has a different pattern of these so-called SNPs. And if we then compare this pattern of SNPs between normal people and those having a prevalence for certain diseases, then we can identify genetic patterns that can be associated with the earlier outbreak of a certain disease. So this is the idea behind it. Works very well for so-called monogenetic diseases, that are diseases that are caused by one single protein that is somehow impaired in its structure and function. But this, of course, can be done without knowing the complete genome. It doesn't work so well for the, let's say, epidemic diseases like atherosclerosis or diabetes. So they have identified many different genetic markers, but it's not clear how they really can predict, precisely predict the onset of a certain disease. So looking just only at the sequence of DNA, the enthusiasm of the first days has somehow vanished, disappeared. So Francis Collins stated 10 years later, after the full sequencing of the genome, that it's fairly to say that the Human Genome Project has not yet directly affected the health care of most individuals. And there are also many critical voices regarding the value of this sequence information alone. So the consequence of this was we have to go along this line of information flow from the DNA down to the phenotype. We have really to understand how the genes translate into the mRNA, these into proteins, these in organic molecules, and in the organelles, as I have stated it before. So there is no other way to understand how a disease may progress and what is the cause for a higher susceptibility. How can we cure a disease that requires to know exactly what happens at each stage of this process from DNA to phenotype. And all this is not new, because I have indicated in blue the fields that traditionally deal with a certain step in this transmission of information. So DNA translation into proteins, that is a field of molecular biology, genetics, and somehow also biophysics. The chemistry of the small organic molecules, that is a domain of biochemistry. Organelles and the cell as a whole, this is a field of cell biology. And if we go up to the organs and tissues, then physiology, anatomy, and medicine are the typical fields dealing with that. So the new thing is that systems biology, and that was the initial question, what is the reason for this new field? That systems biology somehow tries to integrate all these disciplines and to translate step by step in one overall process the genotype, so the genetic information, into the phenotype. So I think that this term molecular systems biology is not well chosen. I would call it integrative biology. Maybe it's a better term, because it's really integrative. It tries to pick up the results of these disciplines and to put them together into an overall picture. And what is new, that this is done not for a single gene, not for a single protein. This is done for all molecules of the cell at the same time. So this field aims at identifying all molecules that are involved in a certain phenotypic property. So the transcriptome, that means the total set of mRNAs, encompasses at least 10,000 different species in the cell. Proteins, the proteome, this is the same size, and the so-called metabolome, that means the set of all organic molecules that we can identify in a cell, is also of the order of 10,000. And all these molecules and their interaction are responsible for what we call the phenotype. And systems biology wants to understand how the interaction among these molecules finally produces a phenotype. So this is the ambitious goal. And the first step is, of course, then to translate the gene to understand how the gene is expressed, how it is translated, transcribed into an mRNA. And maybe at the same time when the genome project started, there was also the beginning of the development of a new technique that meanwhile is present in all laboratories, molecular biology laboratories, as the so-called microarray technique. So the idea is that very small solid state chips, silicon chips, are covered with nucleotides, having a length of 15 up to 20, 30 nucleotides, each of them capable of binding a certain type of RNA molecule that can be isolated from the cell. So this exploits the pairing, the well-known base pairing mechanism, and having 30 nucleotides that can complementary bind a piece of RNA that is enough to bind the RNA, the cellular RNA, on this probe. And one chip may contain up to 10 to the 7 different probes. And the RNA that is taken from the cell, it can be either the mRNA directly, or it can be first prescribed back to the so-called cDNA, and then the cDNA can be used, is labeled with a moiety which can couple to chromophore. So this allows then to quantify which amount of a given RNA molecule has bound to the chip, to which probe of the chip and which amount. So it is a semi-quantitative method to measure the expression level of a gene. That means exactly to measure the mRNA content of a cell. And that can be done for all genes of the cell simultaneously on one chip. So you can measure with this technique the cellular level of 30,000 the activity of 30,000 different genes on one chip. And here this is one of the first chips that had been developed at the end of the last century. It is a microarray having a size of 18 times 18 millimeter, and the chips, the fluorescent and intensity of the various mRNA is then scanned by confocal microscopy. One can label, in principle, we can label these RNAs in 2 different colors in order to distinguish gene activity, for example, at 2 different time points. This was a pioneering work of De Risi and co-workers, the Brown Group, in 1997. The invention of this microarray technique. Here I show you an example from the same publication where they have measured the mRNA level of yeast cells at different time points, and the experiment here was to cultivate yeast cells under conditions of glucose depletion. So there was a permanent decrease in the external concentration of glucose in the medium, so that the yeast cells, which normally have a very high appetite for glucose, that they have to use an alternative substrate for their energy production, and that is the ethanol that is produced by the yeast cells. So in principle, they followed by this technique what are the changes in gene activity in a yeast cell induced by switching from glucose to ethanol. And for those who are a little bit familiar with metabolism of the cell, this part here of that reaction scheme is glycolysis. This is a reaction pathway converting glucose into lactate and pyruvate, into smaller organic molecules. And this is basically the so-called citric acid cycle, which is the metabolic system which finally oxidizes pyruvate, also ethanol, to carbon dioxide, and is coupled to the respiratory chain, which then produces the cellular ATP. And the coloring here indicates that in the moment when we remove the glucose from the medium, there is a decline in the activity of all these glycolytic enzymes, and there's an increase in the, I would say, in the mRNA, to be precise. The mRNA of those enzymes are being settled in the tricarbonic acid cycle, citric acid cycle. So the cells do what we would call a certain economization, in that they remove those parts which are not required anymore, and they then invest more gene activity in the pathway which now comes into operation, that is to use the ethanol in the citric acid cycle as the alternative substrate for energy production. So what is remarkable here in this experiment that we see these changes in the gene expression at almost every enzymatic step. That was in contrast to what we believed up to this point. So in the 80s, we have told our students there are only a few genes which can be regulated, key genes, or the rest is more or less constant. That is not true. We see there is a concerted activation and inactivation of all metabolic genes, at least contained in this fraction of the metabolic system. So again, the question here was, what can be predicted based on mRNA profiles, transcript profiles? And that was a time when they started to compare the expression profile of cells and tissues taken from different donors, normal ones, and those having a certain disease. This is an example in a very early publication where they have compared the mRNA profile of normal cells, normal skin cells, and melanoma. You know, this is a cancer of the skin, and they have analyzed different types of melanoma cells, differing in their invasiveness, degree of invasiveness, and this is then the pattern of those genes which they observe to be most significantly altered in case of this cancer. And what is interesting is here that they were able to provide a very high resolution of these profiles for different types of melanoma. So, for example, it is seen here that one of these genes which codes for integrin. Integrin is a transmembrane protein that is needed for the cell-cell contacts and contacts of the cell with the external extracellular matrix. That this is consistently over or underexpressed in certain subtypes of these melanoma cells. So, and they concluded these cells should differ in their proliferation rate because integrin is required to fix the cell in the tissue. And this can be really proved that is true. So that these profiles give indeed some valuable information regarding the expected phenotype of the cancer cell. That was a time when bioinformatics as a new field entered biology as a very important discipline because these microarrays, they provide a lot of data, more than 100,000 data points per chip. They have to be stored in databases, and then you need statistical methods to retrieve relevant information because there's a lot of noise in these data, which you can imagine because its binding of RNA to the probes is not highly specific, so there is noise which has to be eliminated, so that requires some sophisticated statistical methods. So bioinformatics became an inherent and very important partner in this field. So as I have already mentioned, the advantage of using transcript profiling compared to the conventional diagnostic test is that only based on morphological criteria it's sometimes very difficult to classify a certain cancer type. But the multi-gene expression tests that allow a much more detailed separation into various phenotypes, which possible consequences for their further treatment. So the next step in this along this line of information flow was then to develop methods to analyze the whole proteome of the cell, to make it quantitative. And there are activities mostly in Sweden, Swedish groups, that try to establish a so-called protein atlas, that means all proteins of the cell. And the technique is very, I would say, elegant. They can, first of all, produce from the known sequence of the protein only a small fragment that is enough to elicit an immune response. So then an antibody can be produced against this protein, and this antibody is then purified and labeled, and using this antibody you can virtually identify each individual protein of the cell. So this is really a great work, a tedious work, but they have now the technique to quantitatively determine the amount of each protein in a living cell. And in this publication, these authors did a huge work. They have analyzed almost 5,000 protein profiles in 48 different human tissues and 45 human cell lines. And what they then discovered is that there is a surprisingly high fraction of proteins that are expressed in almost all cells simultaneously. So more than 60% of all proteins can be found in all these different cells and tissues. And they found only a very low fraction of about 2% proteins that are specific for a single cell type. So the conclusion was the specific phenotype of a cell is mostly determined by the abundance of the protein, not by the diversity of the protein spectrum. At the bottom here of this picture you see some examples for proteins that they could identify only in one specific tissue. Then the question came up, is the dogma of molecular biology, is it really valid? So we would expect the mRNA, the amount of mRNA determines the amount of protein that we find in the cell, if we think in that simple terms. And in order to check whether this is true, they have analyzed in 23 different human cell lines more than 1,000 genes and gene products. That means at the same time they have measured the mRNA content of the cell and the protein content pairwise, gene and protein. And then they have studied the correlation. How well does the level of the mRNA really correlate with the abundance of the protein? And this bar diagram shows the correlation coefficient. And you see the correlation in many cases is negative, which is really remarkable. That means the mRNA level is inversely proportional to the protein content, just in a clear contradiction to the central dogma. And the majority of these pairs show a very poor correlation between mRNA and protein level, which is not higher than 0.4. So for predictive purposes, this is nothing. One of the reasons why this poor correlation was found is that there is what we call post-transcriptional regulation in a cell, which after transcription of the mRNA can drastically modify the efficiency of this messenger. For example, there are proteins that may bind to the mRNA to the 5' cap region And binding to this region, they may prevent binding to the ribosome. So then that prevents translation into a protein. So we may have a very high level of an mRNA, but all these molecules are masked by an inhibitor, which is bound to the 5' site. So it's not translated. You don't see any protein. And conversely, there are also stabilizers, other proteins that may bind to the 3' end of the messenger RNA, and because the degradation of the mRNA occurs at the 3' end, where the nucleases remove the nucleotides, that prevents degradation of the messenger. So you may have the situation where the concentration of the messenger RNA is extremely low. It's undetectable with a microarray, but you have a very high abundance of the related protein in the cell. And that is true for many housekeeping proteins. Housekeeping, that means proteins that are always active in the cell. For example, hexokinase, which is a glycolytic enzyme. If you look at the mRNA, it is so small that it cannot be detected. But the protein level of the hexokinase is fairly high. So this is one reason that may account for this discordance between mRNA level and protein level of the cell. That was, again, I have to say, disappointing to see that even though we know the mRNA, we cannot reliably predict what is the protein amount in the cell from the transcript profile. And to complete this more or less frustrating part, then the next technique that conquered this field was mass spectroscopy, which allows to identify small organic molecules with a very high precision. So the idea is to ionize these molecules, and then if they carry a charge, they fly in an electric field. And from physics, one knows that this path is influenced by charge and mass. It is a mass over charge ratio, which is then the characteristics of each of these molecules, and measuring the mass over charge ratio gives then the information on the exact molecular mass of the protein, and you can identify hundreds of these small molecules at the same time using mass spectroscopical methods. So then comparing the amount of enzyme and the messenger RNA in the cell, and relating it to the flux rate, that is the reaction rate that is catalyzed by this enzyme, you see from the scattergram, again, the messenger RNA level of the enzyme, the protein level of the enzyme, they both do not correlate well with the activity of the enzyme in the cell. So you cannot conclude what is the reaction rate of the enzyme knowing the messenger RNA content and the protein content. So after having briefly explained these what we call high throughput technologies that are normally used to explore the molecular composition in a cell-wide range, the conclusion is that these methods, that they are able to make molecular photographs of the cell. So you can make snapshots. You can measure all the mRNAs, the proteins, and also the organic molecules, and you can do it several times, one after the other. But the key point is, this is not predictive. Having 100 of these photographs, you don't know how will look like the next photograph. You do it again. And the reason for this is that these photographs, these molecular pictures, they are the result of a dynamic process, and this dynamic process in the cell is determined by the molecular interactions. these are two examples of those interaction networks. One is fairly old. This is an insert from a metabolic map, which you can find at the door of many laboratories. So well-known metabolic map. This is a scheme that shows all reactions in the cells and the reaction partners. So the typical element of such a network is a chemical reaction where molecules react with each other to form products. A, B react to form C and D, and C forms E. Each of these reactions is catalyzed by a specific enzyme. And the problem is that the molecules may interfere with the activity of the catalyzing enzymes. So for example, E, the metabolite E, can feed back to the activity of enzyme E1, which we call an allosteric interaction. So in this case, E is a negative effector of enzyme E1. And moreover, enzymes can be phosphorylated or dephosphorylated, and they differ in the activity whether they are in the phosphorylated or in the dephosphorylated state. So you see, the reaction network doesn't tell you a lot about these types of regulation, these allosteric effects as well as the phosphorylation-dephosphorylation reactions. In the so-called signaling networks, that are protein networks that are involved in transmitting an external signal into the cell, the typical situation is that upon binding to a receptor, a target molecule in the cell and protein is phosphorylated. That in turn may act as a kinase phosphorylating other proteins, so there is a cascade of phosphorylations inside the cell. These can even form complexes, and at the end, a phosphorylated protein, which can be the 10s, the 15s, the 20s in this cascade of phosphorylations, enters the DNA as a transcription factor. So we have to face a situation where all these molecules that we can trace by these high throughput methods are interconnected by these types of molecular interactions. So if we want to go one step further and to understand the dynamics of the cell and to explain these snapshots in a mechanistic way, then we have to go on to identify all these molecular interactions and to quantify these interactions. Here's an example showing that it's a very cumbersome procedure. So knowing, for example, a certain protein that binds to an RNA. It can bind, it can be released, so that is an equilibrium process. So you may write down an equilibrium equation for us, a mass balance equation for this process. And then you have to go on to measure it. So to have somehow to isolate this messenger, and then you have to make the protein. That can be done, for example, expression of this process in a bacterium. And then it has to be followed how that binds to the RNA. And here that was done in a Gel-Shift assay. So because you can then identify the complex, the bound messenger RNA to the protein in the gel, that gives a dependency binding versus concentration of the protein. And finally, from this information, experimental information, one has to estimate the parameters in the binding model. This is a work for a postdoc. It takes 2-3 years for one molecular interaction. It's much more time-consuming as 100 microarrays. And I guess many people think there is no way to proceed in this manner to identify molecular interactions on this level, step by step, these millions of interactions, and then to put this together into a dynamic picture. Because the title of my talk was also to mention the important role of imaging procedures. This is a good point to show that indeed these imaging procedures have contributed a lot to identify molecular interactions, dynamic interactions in the cell. Here it is confocal fluorescence microscopy that has to be mentioned. It is a method that now allows to trace interactions in a time-resolved manner. I give 3 examples. One is the so-called FRET technology, which is that 2 molecules which are supposed to interact are labeled by a fluorescent dye, and one is fluorescent and the other is not. And if they approximate below a critical distance, then an energy transfer occurs between molecule A to molecule B to the chromophore, and then it is excited and it gives a fluorescent signal. So the fluorescent signal then of the second molecule is indicative for an interaction with molecule A. And this is a very elegant method to determine molecular interactions in vivo, to see whether the hypothesis on a possible interaction of 2 proteins or 2 other molecules is actually true in vivo. A second very important application of this method is the fluorescence recovery photobleaching method, that is which has helped to estimate the fusion rates of proteins inside the cell, and in particular in the membrane. So then the molecule under interest is labeled again with a fluorescent dye, and which is self-fluorescent, and then upon illumination by the laser beam, the fluorescence is bleached for a moment, so this spot of the cell surface is dark, and then owing to diffusion, the labeled fluorescent molecules enter this dark region. So this is a recovery of fluorescence in this spot. And from the dynamics of this process, which is shown here, one can estimate the diffusion time, the diffusion constant of this molecule. And then a third application, which I find very fascinating, is you can really trace the movement of molecules in a cell. This is shown here. This is the movement of mitochondria along microtubules. Microtubules that span from, this is a neuronal cell, this is a soma, this is an axon, and this is a microtubule. And the mitochondria are transported from the soma of the cell to the axonal termini, where they are required for ATP production, because the most of the energy that is used by a neuron is indeed not used for the action potential generation. It is used for the release of neurotransmitters in the axonal termini and the reuptake of these neurotransmitters into vesicles. And by this technique, one can really measure what is the rate for the movement of mitochondria along these microtubules. A fascinating technique which has helped a lot to understand the spatial dynamics in the cell. So the ultimate goal of systems biology is to set up an in silico cell model, because the mathematical model is that's the highest level of understanding what one can present. Maybe you know this famous sentence of Karl Marx, the famous philosopher and social philosopher, who stated that science has reached its highest level if it is capable of using mathematics. And therefore, it is the goal of systems biology to set up mathematical models of the whole cell. And maybe you are aware of projects, international projects, Dutch groups, German groups, Japanese groups, and even American groups claiming that they're on the way to develop whole cell mathematical models. So in essence, this model would mean to put a grid, a three-dimensional grid on the cell, and then to define the number of particles in each of these grid points, and then to predict, using a mathematical model, what is the number of molecules of a specific sort at any time in a cell. This is a dream. This is what some people have in mind, saying we want to set up an in silico cell model. The main issue is that up to now we do not know how the processes leading to the formation of organelles in a cell, so the spatial arrangement of molecules, can be really described by mathematical models. So there are no reliable models doing this. So therefore, normally the cell is decomposed into certain compartments. Each of these compartments is thought to be well mixed, so what we can assume we have an more or less homogeneous distribution of molecules inside each of these compartments. And then these compartments, as the ER, the mitochondria, the membrane, and so forth, they are connected by transport processes. This is a type of model that is commonly used in systems biology to refrain from looking at the real spatial situation in a cell. And then the goal is determine the concentration of a certain molecule species inside a given compartment. So the mathematical procedure that is used in 90% of all cases are models that are derived from chemical kinetics. So these are differential equation systems. The variables are the concentrations of the proteins, the mRNA molecules, the organic molecules of all molecules of the cell, and the processes connecting these interacting molecules are described by so-called rate equations. And as I have shown before with this example of protein mRNA interaction, it takes some time to set up the rate equation for a single process, but once you have done this, then you can put it into this equation system and you can solve it, and then it gives you a deterministic description of the cell dynamics. So we then have to solve differential equation systems of this type, where the time-dependent change of a metabolite is given as a result of the processes which can be decomposed into chemical reactions and membrane transport processes. I think it's not necessary to show more details here. This is a rather old technique, I have to say. At least in metabolic research, the first type of these models was published in the 70s. Look at this. This is a Garfinkel model. This is a physiological model. It describes the energy metabolism of the heart, and it was used to quantify changes in the metabolic rates under conditions of ischemia. And this model was composed of many, many biochemical reactions, each of them characterized by rate equation, then a large system of differential equation. I cannot remember how many, but at least 100 was solved to predict the time course of these metabolites, and that can be compared with the experiment. So the idea of a mathematical model describing cellular processes is fairly old. It's not an invention of systems biology. That is called, sometimes pejoratively, as reductionist approach. So people prefer these high throughput technologies. They say, okay, you can understand well a very small defined system. You can even set up a mathematical model giving exact predictions that can be then checked experimentally. But you can never describe in this way the whole cell. This is restricted to small systems. On the other hand, the high throughput approach, as I have illuminated in the first part of my presentation, cannot really be predictive. It can give you some information on what happens, so a pattern of molecular events, but it is of more or less, it is not predictive. So we have two, let's say, opposing subfields in systems biology at the moment. This is on one hand this data-driven high throughput approach, giving these photographs, and we have still, I would say still, because it is fairly old, we have this other branch, which is called reductionist, setting up relatively small mathematical models for well-defined systems where you know each interaction very precisely. So the question is how to integrate these two approaches, or can they be integrated? And my answer is yes, there are ways. One is the high throughput analysis is explorative, so it gives you at least a key where you have a lamp, where you have to look inside the cell for those processes which are most relevant for a certain phenomenon, because they can identify the most significant changes in mRNA, proteins, and organic molecules at the same time. But then there is no alternative than to go into the depths and to look what really happens behind this. And then step by step, in a more or less time-consuming process, then identify all these molecular interactions, as I've explained it before. Another way I personally prefer is to have a clear hypothesis. We should learn to more thinking in terms of hypothesis, not in the way, let's the data speak, we will see something, fishing in a lake, we will see what we can capture. Maybe we can really identify what fish we have on angle. Maybe not. No, we should understand, develop a feeling. What are the strategies of the cell and how they are reflected, these strategies reflected in the molecular interactions. It was Weinberg, one of the famous persons in cancer research, who then published this article where he clearly says hypotheses first. He has a very critical view what all these high throughput techniques had really provided in the last 10 years. All this money that had been invested. Was it really worth to spend this on the other hand, to stop many smaller projects which looked on the details of the process that finally results in cancer progression. Let me give, at the end of my presentation, what I mean by hypothesis-driven. This is, although we are requested not to refer to our own work, this is the only reference I make. This was a work, it was 10 years ago. That was just when the Risi experiment had been published, where we thought, what could be the strategy behind temporal gene expression? Why this change in the activity of metabolic enzymes that the Risi has observed in the yeast cell? You remember this picture where glycolysis was dropping down and the citric acid cycle was activated. And our hypothesis was, this is a way of a cell to economize usage of proteins, given that the total amount of proteins in the cell is limited for several reasons, which I don't want to explain here. Then the cell has to distribute proteins in a way that they are optimal for functioning of the cell. So if glucose is there, then it makes sense to put a lot of enzymes to the glycolytic pathway. If glucose is not present, then it's more gives more sense to spend this protein amount to the citric acid cycle. So our hypothesis was simply the temporal expression of genes can be accounted for by an economic usage of proteins. And we have done this based on a mathematical model of yeast metabolism, and based on this optimization principle, we have predicted what should be the optimal gene expression. And indeed, that was in good agreement with observed expression profiles. And later on, picking up this idea of an economic usage, so the strategy of the cell to use protein, the limited amount of protein in the best way, picking up this idea, the group of Uri Alan has really verified this experimentally. They have traced the expression of proteins in a synthesizing pathway and found it is in agreement with this hypothesis. So having a hypothesis about the functioning of a system can really help to better rationalize what is the principle that governs the dynamics of molecules in the cell. So with this I want to end up and I'll summarize my presentation in the following way, that systems biology is an interdisciplinary research field comprising scientists from various fields, physicists, mathematicians, biochemists, cell biologists, and so forth. And it aims at understanding how molecular interactions may translate in phenotypic properties. It uses, and that is a typical characteristics, high throughput technologies, including imaging techniques to quantify in a cell-wide range dynamic changes. It needs bioinformatics to manage huge data sets and to get the information out from these data sets. And it attempts to rigorously use mathematical models to be quantitative and predictive. And my personal conclusion is it can only be successful if it is able to integrate the discovery-driven and the hypothesis-driven approaches. Thank you for your attention.